Hey everybody, welcome back to General Biochemistry. We are still in chapter 28. This is the last video of the semester, so clap it up. You got that far. I'm doing like the almost clap hands. We're going to pick up with section 3 of chapter 28, where we left off. We're going to be talking about more of these regulatory proteins and how they work to regulate gene expression. So here we go. Regulation of gene expression in eukaryotes. Remember, we skipped over bacteria for the most part. So when we were talking about positive versus negative regulation, we said that eukaryotes primarily use positive regulation, meaning that we use activators versus repressors. We're going to talk about that more now. Initiation actually depends on multiple activator proteins, not RNA polymerase affinity. So that's one big difference with, eukaryotes and bacteria. And just like with DNA and RNA metabolism, and protein metabolism, be thinking about some of the differences that we've talked about between bacteria and eukaryotes. Even though we didn't cover everything in nitty-gritty detail about bacteria, there are some, kind of global ideas that carry forward from those chapters to now. Chromatin, which is the structure that your chromosomes are in, it's the DNA and the RNA and the proteins all wrapped up in a package. Chromatin renders most promoters inaccessible. So repressors would be redundant in this case. If you can't access the DNA, you don't have to have a repressor bound. Why go through the trouble of making a protein to repress something that you can't access in the first place? Combinatorial control gives you precise positive control, and negative regulation in eukaryotes typically involves those long non-coding RNAs. So let's talk about the advantages of using combinatorial control. Let's say that you have two regulatory gene families, and each of those two families has 3 members that can form dimers. If you do out all the possibilities, you've got 36 different regulatory combinations that are possible. Now just think about how you can have 36 different activation states, because that's what we're really talking about here. That each of these combinations gives you a slightly different amount of transcriptional activity. And that gives you a lot of room to increase or decrease transcription on the fly and have a very, very slight, subtle increase or decrease in transcription, which then propagates forward to the gene product, whether it's RNA only, or if you're making a product like protein at the end. DNA binding activators. We're talking about some of the different DNA binding domains, but now we're going to talk more about these for eukaryotes. So DNA binding activators and co-activators facilitate the assembly of all those basal transcription factors that we talked about when we were talking about RNA polymerase II in eukaryotes. So many Pol2 promoters, not all, but many of them, have a TATA box and an initiator sequence. And there's a standard spacing between the two. But there can also be additional regulatory sequences that are bound by transcriptional activators to increase transcription. Enhancers, which is what we talked about again in the last video, are present in higher eukaryotes like us, mammals. And they can be located anywhere, because those enhancer regions don't have to be physically close. We can do some chromatin remodeling, we can do some bending of DNA, and bring things together. In yeast, there are upstream activator sequences, that's abbreviated UAS. It must be upstream, and with yeast, it's generally within a few hundred base pairs of the promoter. So let's talk about the types of proteins that are required for pol-2 in all of its glory to bind. We've got all the transcription activators, which bind to enhancers, or in yeast, the upstream activator sequence, and they facilitate transcription. We also need the architectural regulators, which facilitate DNA looping. Then we've got the chromatin modification and remodeling proteins. We talked a little bit about these, and we're going to continue our conversation. We also have the co-activators that communicate between the activators and the pol-2 complex and all of the basal transcription factors. And these co-activators play a direct role in the assembly of the PIC, the pre-initiation complex. Then we have the basal transcription factors, which are the general transcription factors that were introduced back when we talked about RNA metabolism, and then we talked about how, they all come together and make the pre-initiation complex and yada yada. So that was just one layer that we covered in that chapter, believe it or not. So let's talk through the activation of transcription. We're looking at yeast because there's a UAS here versus an enhancer region. So remember that difference. There's lots of different ways that you can have regulation. One way is that you can have a long non-coding RNA bind to the protein that is bound at the UAS, or it can competitively bind with this particular sequence to make it so that you actually have silencing of this gene. Now, let's say that long non-coding RNA is gone. Then maybe that allows for mediator, which is one of those co-activators, to bind the activator and be the bridge between the activator and the pol-2 holo complex. These HMG proteins, which we're going to go through all of these things in text, but I'm going through the picture a little bit first, and then you'll get the words. So the HMG proteins, these are the remodeling proteins. They're the ones that help to make the DNA loop and bring the activator, co-activator, and the pol-2 complex together. Then you also have transcription activators and co-activators and things of that nature on other sequences. So it's not necessarily just one thing, just the UAS and yeast, or just transcription activators. But we're talking about all of these things coming together. These transcription activators and other binding sites can also interact with mediator, a co-activator, and other transcription factors that are associated with pol-2. Once all of this happens and you have this looped DNA that's got an assembled pre-initiation complex, then pol-2 is ready to roll and you can make that messenger RNA. You can also have repression here. So there are multiple ways. One would be the long non-coding RNA, like I talked about before, which they can actually activate as well. It just depends on what the RNA is. But they can have an activating role or repressing role. It just depends on which gene or gene cluster. So you can have in yeast, again, a repressor or a long non-coding RNA that binds to an activator or binds to a repressor or something of that nature to make sure that, hey, you guys are not going to talk to each other. Mediator, get out of here. So the repressor might bind mediator and cause it to not interact with pol-2. There are lots of different ways that you can imagine that these proteins can interact and cause some kind of a glitch in the system to where you can't have all of the interactions necessary to bring forth transcription. So this is just a very generic example, and this is one of the figures that you can use to kind of talk through the different ways that you can envision repression of transcription, you can use the other figure to talk through the different ways that you can activate transcription. So this isn't an end-all be-all, but think of it more as a way to look at some of the different varieties of how transcription is regulated. So let's talk about some of these in a little bit more detail. Let's start with transcription activators. So the activator requirements are really different from promoter to promoter. We could be talking about activating transcription at hundreds of promoters, or you could have an activator that's very specific for a few promoters. They might be sensitive to signal molecule binding, which we'll talk about some of those at the end. Maybe it binds to an enhancer that's really far away from the data box. So you can see there's a lot of variation here. And this is really just to get you thinking about the possibilities so that, again, when we do case studies, you'll be able to kind of hit the ground running and already have the foundation for how the system might work. Activators might also bind both DNA and RNA. Be thinking about the types of motifs that you can use in those types of proteins. And their function may be affected by long non-coding RNAs, either activating or repressing. The architectural regulators, those are the ones that were kind of the half moons. Those facilitate DNA looping and allow the activators to function at a distance. So just like we were talking about in bacteria, when you loop the DNA, you kind of bring the things together that normally wouldn't talk to each other. These are very abundant in chromatin, and they bind to DNA with very limited specificity. So they may have DNA binding regions, but they don't actually bind a specific sequence. It's just like, hey, there's some DNA. I'm going to stick to that. And that's it. HMG proteins, which stand for high mobility group proteins, play a really big role in chromatin remodeling and activating transcription. So you'll always see those as part of the mix. This is just a figure of the HMG domain and how it's kind of like a hand just cupped around the DNA. There's no real specific spots where this domain is poking into the DNA and probing, the major groove for any specific binding sites. It's just like, hey, DNA, I like it. I'm going to stay. Then we've got co-activator protein complexes. Now, I showed mediator, which is a complex, and it's a major eukaryotic co-activator. It's in yeast. It's in humans. 25, 30 different proteins. Lots of them are very highly conserved. Makes sense. It binds to the C-terminal domain of the largest subunit of POL2. And remember, POL2, that CTD, very, very active for phosphorylation, very active and modular in terms of the things that bind to it. And all the things that bind to it kind of tell you where that polymerase is in the process of making, the transcript. And there's just lots of regulation here. Mediator is actually required for basal and regulated transcription. And it facilitates the effects of those transcription activators on POL2. So mediator is literally the center of everything. And it's a co-activator that will interact with activators and transcription factors and POL2 and all of the things. Then we have the TATA binding protein, or TBP, and all the basal transcription factors, which we already talked about these at length in an earlier lecture. So if that's a little bit fuzzy still, go back and revisit that. TBP is the first component to bind in the assembly of the pre-initiation complex, usually. And then you bring in TF2D and then all the other factors. The basal transcription factors they make up the minimal pick, the pre-initiation complex, along with POL2. It's insufficient to initiate transcription in vivo, which means in the cell, but you might be able to get something to work on the benchtop. So, as you can see, with all of these different layers of complexity and regulation, transcription is really a highly choreographed dance. Let's talk through all the choreography of what happens. So the activators bind to the DNA. They're at their enhancer region hanging out. Activators will recruit the remodeling complexes and a co-activator. We're saying mediator here, but there are others. When we talk about the remodeling complexes, we're talking about like the SWI/SNF complexes and things like that, where you come in and you boot out some nucleosomes and you make that DNA more accessible. That's just one example. So now, if you're following along with the pictures, we've got our activators bound. We've got our remodeling complexes and our co-activator. Mediator will facilitate the binding of all the other required components. So we're looking at all of those transcription factors. We're looking at any other specialized things that might be needed for transcription at that particular promoter. All of those things are binding, including the polymerase, of course. And then once you have all of those things binding in the order that they bind in, then you've got phosphorylation of that C-terminal domain of pol 2, and you kick off transcription. These are the genes that are part of galactose metabolism in yeast that are subject to both positive and negative regulation. This is not at all for you to memorize, but just for you to see kind of where the different genes are on the chromosomes, how big they are, how small they are, relative expression, and things of that nature, just to get an idea of what regulated genes look like, what their regulatory genes look like, and kind of have an idea of their comparison. We are going to talk about these genes and give you a bit of an example of a lower eukaryote and regulation. So the promoters for the gal genes, which these are just the genes that are necessary for yeast to use galactose as a source of energy. So we've got the TATA box, we've got initiator sequences, upstream activator sequence. This one is specific for galactose and is recognized by the transcription activator gal4 protein, which, as a reminder for yeast, anytime you see a P at the end of something, it's capital letter, lowercase letters, a number, and then the letter P, we're talking about a protein. Whenever you see italics, like the gal genes, then you know that you're talking about a gene. So for this regulation, we talked about the UAS, right? So that's over here. Gal80 and Gal4 proteins form a complex and hang out on that UAS. The Gal4 protein is prevented from functioning as an activator when it's in a complex with Gal80P alone. When galactose is present, then we have the binding of Gal3P. That's this little blue guy here. And it will bind galactose, and then it will go and find this Gal4, Gal80 complex and bind it in such a way that it allows Gal4 to function as an activator at the Gal promoters. We're going to recruit additional things like the Saga complex, which is a remodeling complex in yeast, and SWI/SNF complex, again, more remodeling. And we're also going to bring mediator along for the ride. Once you bring mediator in, you know what happens. You're going to recruit all of those other proteins, the transcription factors, and pol 2, and you're going to initiate transcription. And once you do that, you'll have all the genes turned on, that are necessary for using galactose as a source of energy. So it's very, very straightforward. Well, I say very straightforward, but as straightforward as it gets when it comes to eukaryotes. Yeast are kind of the golden child for looking at things like this. So Gal4 is an activator. Things about Gal4 that are interesting that should make sense. Zinc finger-like structure in the DNA binding domain. We talked about all the different, not all, but several motifs that are common with DNA binding domains. It functions as a homodimer. Lots of these activators do. We talked about the lac repressor in bacteria. It's a little bit different. It's a tetramer. It's two dimers that come together. But Gal4P is a little bit more traditional. It functions as a homodimer like most others. It binds to a palindromic DNA sequence that is the UAS. And it also has an acidic activation domain. And that acidic domain is actually critical for its function. If you mutate it or get rid of it, it's not going to act as an activator anymore. So why did we look at Gal4? Well, it exhibits the fact that transcription activators usually have a modular structure. So there's the DNA binding domain, and there's some kind of an activation domain that says, hey, get started being an activator. Remember that Gal4P, when it was in complex with Gal80P, it didn't act as an activator. But then you have a conformational change when Gal3P binds. Suddenly it does. Think about what that might do for an activation domain. Maybe it allows it to become accessible so that it can interact with mediator. There are several other activators that yeast have, and we'll talk about two of them, SP1 and CTF1. So SP1 is a transcription factor for lots of genes in higher eukaryotes. Usually binds near the TATA box, and the DNA binding domain is near the C terminus, and it's got 3 zinc fingers. Get my highlighter out. So those 3 zinc fingers are near the C terminus. You have to have them to bind the DNA. It's also got a couple of glutamine-rich domains that function in activation. There are lots of activator proteins that also have these glutamine-rich domains, and they're literally a quarter glutamine. That's a lot of glutamine in one little motif. The CTF1 binds to a particular sequence, CCAAT, and it's an activator. It has a binding domain that has a lot of basic amino acid residues, and the region is probably an alpha helix. Being that it says probably, you can guess that it's not 100% known. This protein also has a proline-rich activation domain, which proline is interesting, and it accounts for more than 20% of the residues in that domain. So that is its activating domain. So how do you know that you can, how do you know that these transcriptional activators are modular? The reason is you can do experiments to actually swap domains. Think about how you might do that, do all of the cloning to make some of these constructs, thinking about chapter 9. So all of the ways to manipulate DNA and make protein expression plasmids and kind of cut and paste. Think about that with these experiments, because that's exactly what they were doing. So activation and DNA binding domains of these regulatory proteins usually act independently. So there may be some linker regions in between the domains, but you can kind of cut them apart and then paste them onto something else, and they'll still work as an activator. So you can use genetic engineering techniques to join the activation domain of one protein with the DNA binding domain of another. So, for example, if you had the DNA binding domain of SP1 and you add to it the activation domain of CTF1, then you would get a protein that recognizes the same sequence that SP1 recognizes. And it would activate using those same sequences. And then it would recruit mediator with that proline-rich domain. And you can still get all of the transcriptional activation using this chimera of a protein. So you could, in theory, use this hybrid to activate proteins that normally wouldn't be activated with SP1. Or you could use this to, let's say you flip-flop it and you use the CTF1 binding domain and the SP1 activation domain. So there are lots of things that you can do to figure out what parts of a protein are necessary for activation, DNA binding, etc. So eukaryotic gene expression can also be regulated by intracellular and intracellular signals. We have to talk about hormones and hormone signaling. It's a big thing in higher eukaryotes, of course. Hormone receptor complexes will bind to DNA sequences that are called hormone response elements, or HREs. And these receptors act as transcription activators and recruit the co-activators and POL2 and all the transcription machinery, just like these other activators that we've been talking about. Hormone receptors, they have a consensus HRE sequence. And each receptor has a different sequence. So the androgen receptor has its own HRE consensus sequence, glucocorticoid, etc. So you can see all of those in table 28-4. They usually have a highly conserved DNA binding domain with 2 zinc fingers. And there's a specific ligand binding region at the C terminus. So this is the basic structure of these hormone receptors. They have to have something that's going to bind to that consensus sequence and something that's going to recognize the ligand that it's supposed to respond to. So this is just your typical steroid hormone receptor. What's shown here is the sequence down at the bottom for the zinc finger is actually for the estrogen receptor, but there are certain things that are conserved. So all the bold letters are conserved residues. So in terms of DNA binding, those are going to be conserved. As we said, there's a transcription activation, which is variable in its sequence and length, but it's the activator part of the receptor. Then there's the DNA binding, which is highly conserved. And then there's the hormone binding region, which is going to vary, right? Because you have to respond to different hormones. But this is the basic makeup of these steroid hormone receptors. Here are some examples. There's a couple of types of signaling that we can talk about. So there's monomeric type 1 receptors. An example of that would be sex hormones, glucocorticoids. They're found in the cytoplasm and they complex with HSP70. So what happens is you have a hormone that gets through into the cytosol and it meets up with its receptor friend and binds. Once that happens, HSP70 dissociates and you now have your receptor hormone complex. Two of those will come together and dimerize, and they'll get imported into the nucleus. The dimerization of those receptors is what exposes that nuclear localization signal, the NLS. And if you remember, we talked about that when we talked about post-translational modification and processing of proteins. So it's that same nuclear localization signal that gets exposed that allows this dimer to enter the nucleus. Once it gets there, it can bind to the hormone, its element, and then it can recruit mediator or whichever other co-activator is necessary, and then help recruit all of the basal transcription factors and POL2 to turn on transcription, of the target gene. You can have another layer of regulation here. Let's say that after a certain time, you need to turn off this response. Well, you can have something else that causes that dimer to dissociate from its elements so that it stops activating transcription. And one thing that can happen is you can have a long non-coding RNA come along and compete with the binding site for that enhancer region. So it'll bind to the dimer so that it gets off of the promoter, and then you turn off your gene. Type 2 receptors are a little bit different. One example of a type 2 receptor is the thyroid hormone receptor. The hormone enters the cell, and it just goes straight through to the nucleus. Doesn't stop for anything. Once it gets to the nucleus, it's going to bind to an HRE and a co-repressor. Once it binds, that co-repressor is going to dissociate, and then all of a sudden the receptor that's present can act as an activator. So you're kind of popping the lid off and letting everything out so that you can have activation of transcription. So now the hormone can bring in the co-activator and POL2 and all the other transcription factors necessary to turn on transcription. And of course, with both of these, you'll make some kind of a product, some kind of a messenger RNA, and then you make a protein, and that protein is going to do something in the cell. So it's going to serve some function that the cell needs. Now, I've been throwing in those long non-coding RNAs all over the place. And they really are all over the place. But let's talk about one specifically. Gas5 is a long non-coding RNA that inhibits transcriptional activity by the glucocorticoid receptor and related receptors. So it directly competes for that receptor binding, because remember, it's just nucleotides, right? So if it looks like the same sequence that the receptor binds in the nucleus, then it can, bind there. Steroid receptor RNA activator, SRA, is a long non-coding RNA that acts as a co-activator. But it doesn't act completely alone. It's part of a ribonucleoprotein complex. But you actually need the RNA for the activation activity, not just the protein alone. So it really is an RNA-centric world out there. I should also mention that Gas5 is a tumor suppressor gene. And so if you don't have expression of Gas5, then that's real bad news bears. And you'll actually see that suppressed Gas5 expression it correlates with worse outcomes and worse cases of cancer. So it really is incredibly important. So regulation can also result from phosphorylation of nuclear transcription factors. Remember that transcription happens in the nucleus, and so all the same rules apply to the enzymes and proteins in the cytosol. Phosphorylation is a very powerful signal that can change how proteins talk to each other, and it can change the structure and function of a protein within itself. Non-steroid hormones bind to cell surface receptors, and that can trigger a whole signaling cascade that leads to phosphorylation of regulatory proteins, and that will regulate its activity. So for example, in the beta adrenergic pathway, the result is that you increase cytosolic cyclic AMP, and that activates protein kinase A. We've heard a lot about PKA, right? It's one of those master regulator proteins. PKA, when it sees that increased cyclic AMP, part of it will dissociate and enter the nucleus, and it will phosphorylate. So the part that dissociates is the catalytic domain. It will enter the nucleus and phosphorylate the CRE binding protein, or CREB. Phosphorylated CREB binds to cyclic AMP response elements and acts as a transcription factor. So this is going to increase gene expression of all of the genes that have a corresponding CRE. This is just one example of how signaling from the outside can tell the cell to amp up gene expression on the inside. Many eukaryotic mRNAs are subject to translational repression as well. So it's not enough that we can modulate how much transcript we make, but we also can control how much protein is made by controlling how many ribosomes are really able to sit down and bind to that mRNA and make a polypeptide. So activating a translationally repressed mRNA in the cytoplasm is actually way quicker than making the transcript fresh and then exporting it to the cytoplasm and then making the protein. So this is actually a tool that the cell uses to kind of have a preloaded cytoplasm that's full of all these mRNAs. They're just silent. They're just waiting for the time to act. There's 4 main mechanisms of translational regulation. Phosphorylation, of course, of translation initiation factors. Binding of translational repressors, which often they'll bind to the 3' untranslated region, the UTR. Binding proteins that disrupt EIF4E and 4G interactions. And remember that they're necessary for kind of helping to bring the RNA around to make the actual competent complex for the ribosomes to start. And then there's RNA-mediated regulation, which is often translational repression. So first, let's talk about the UTR and what happens there. Repressors can bind to specific sites in that 3' UTR, and that will block or slow translation. What happens is these repressors will interrupt the association of the mRNA with the different factors, either the 5' cap or with the poly A binding protein, with EIF4F and 4G. It can pretty much, anything that you can imagine, there's some kind of a repressor for that, okay? In reticulocyte maturation, so we're talking about maturing blood cells, red blood cells. What happens to fully mature these cells is you get rid of the nucleus. And that sounds really crazy, but what happens is there's lots of mRNAs that are deposited in the cytoplasm before the nucleus is destroyed. And all of those mRNAs are just waiting around for the signal to be released out into the wild and make more hemoglobin or do whatever else is necessary to repair the red blood cell and make it, competent for work. The human-controlled repressor, HCR, catalyzes the phosphorylation of EIF2, when reticulocytes are deficient in iron or heme. What's the point in, you know, having all of your transcription factors available when you don't need them? So, if you don't have iron or heme around, you don't really want to be trying to make hemoglobin. You need it. So, it's going to sequester EIF2 and make it unavailable for translation. And then, when heme is available again, you remove that phosphoryl group and you can actually start synthesis of the globin part of hemoglobin. There's also lots of gene silencing that's mediated by what's called RNAi, which stands for RNA interference. MicroRNAs, which is abbreviated miRNAs, they will bind to mRNAs and mediate gene silencing. This is critical in developmental timing in some organisms. It will keep certain messenger RNAs quiet until the right time, and then they can be allowed to make their product, and then that product will go on to aid in the development of the organism. This also protects against invading RNA viruses, particularly in plants, because they don't really have an immune system. So this will quiet viruses, and plants are preloaded with all these different RNAs that actually recognize plant viruses. So it will bind to them and shut them up. The RNAs that are involved in development are sometimes called small temporal RNAs or sTRNAs, and they're just microRNAs that are present very brief amount of time during development. But it's a subset of microRNAs. We didn't cover the dicer complex very much when we talked about RNA processing, so I'm kind of going to gloss over this a little. You don't need to know the details of the dicer mechanism, but just understand overall what it does. So when we're talking about RNAi, we're talking about gene silencing, where a dicer, which is this big complex, will cleave a duplex RNA, meaning it's RNA that is double-stranded, or it could be a single RNA that's got a hairpin structure, so it loops back around and then it will base pair with itself. But dicer will cleave these RNA molecules to create small interfering RNAs or siRNAs. And they can bind to messenger RNA and silence it. So if you have a siRNA that binds to messenger RNA, it will either silence it so that you can't translate, it will do something to the structure so that the ribosome can't bind, all kinds of things. Or it can label this mRNA for destruction. So that's the purpose of RNAi. This has also been harnessed in the lab as a tool for investigating the purpose of RNAs. So you can make a small interfering RNA that targets a particular messenger RNA of a protein you're interested in looking at what happens when you don't have it. And that's a way to look at that and explore it without mutating the organism. So RNA-mediated regulation of gene expression takes a lot of different forms. Kind of a theme here. A lot of different ways that things can go down. Those non-coding RNAs, again, they don't make protein. When they're super long, they're long non-coding RNAs. And we have an example where a long non-coding RNA can stimulate heat shock factor 1 trimerization and DNA binding to activate transcription. We have one that will bind to POL2 to the POL2 transcription elongation factor and repress elongation. There are so many examples of what these RNAs can do. It's kind of crazy. So you don't need to necessarily memorize each of these examples, but more think about the possibilities. So you can have stimulation of transcription, you can have repression of transcription, you can repress elongation, Lots of different things. And all of these RNAs are also subjected to the same kinds of regulation that we've been talking about. So it could be in response to some kind of a stress. It could be in response to a hormone that's secreted. All of these things can stimulate the creation of these non-coding RNAs, which then go on and do a job. So development is controlled by cascades of regulatory proteins. It is a very complex process, and embryonic development requires really tightly coordinated changes in expression. More genes are expressed during early development than any other part of your life cycle. So sea urchins, I don't know if you can even envision what a sea urchin looks like. I had to look it up. I'm not even going to lie. I was like, a sea urchin? What's that? I know the name. But can I conjure a picture in my mind? No. Ask Google. Anyhow, in the oocyte of a sea urchin, you've got 18,000 some odd different messenger RNAs. But in a differentiated tissue, about 6,000. So big difference. One of the main lab animals that are used to investigate development is Drosophila melanogaster. It's the fruit fly. Now, my family loves bananas. My kids will tear bananas up. You leave them in a room with a couple of bunches of banana, you might not even have peels left if they're that hungry. Because of this, I'm very familiar with our old friend, the fruit fly. They love some banana peels, okay? So Drosophila undergo a complete metamorphosis going from that little teeny tiny embryo to an adult. And as part of that journey, they make this kind of elongated blob, but that blob is segmented. And each of those little segments is destined to become a certain body part and have certain appendages. And you can imagine that the head and the tail are going to have different requirements. The head's going to have the eyes and everything else, the mouth. The abdomen's going to be a little bit different. You're going to need wings in there somewhere. So all these different segments are necessary to differentiate into all these different appendages and all their little organs and insides. With Drosophila, we've got an oocyte that needs to mature. Once it finishes maturing, it's going to have all of these messenger RNAs, the maternal mRNAs, and proteins that are deposited by nurse cells and follicle cells during this maturation process. After fertilization, you're going to have a nuclei of an egg that's going to just keep dividing and dividing and dividing. So if you're keeping track, the first bullet point we're talking about here. After fertilization, then we've got this zygote. So fertilization happens here. And then you've got the nuclei just dividing, and they're all sharing the same cytoplasm. So this is kind of 2-ish. And 2. Have to do the circles. It's one of those incomplete things. It's like, you got to finish the book once you start it. I circled the others. Had to circle these 2. So notice how there's no kind of boundaries around those nuclei. They're all just hanging out in the cytoplasm. Well, the next part is where we've got nuclei migration to the periphery. And then you're going to start creating invaginations and creating a monolayer of cells. We've made a blastoderm. And you're going to keep on having that kind of process where you're making all these cells and they're going to go to different places. Within those cells, we're going to have all of these mRNAs and proteins. And the proteins that cause the surrounding tissue to take a particular shape or structure is called a morphogen. And these function through changes in local concentration or activity. So you can imagine having a concentration of a protein on one end that's supposed to be the head, and then as you get closer and closer to the tail, you don't see that protein anymore. Morphogens are the products of pattern regulating genes. So we've got maternal genes, segmentation genes, and homeotic genes. For all of this fly development stuff, this is not a developmental biology class. This is really a way to kind of bring all of the things we've been talking about with regulation to a case study. So you should understand in general what's going on here with this case study, but that's really because you should understand how these different players come together to work to develop an organism. So this is less about memorizing and more about using what you've learned in chapter 28 thus far to understand development. So the maternal genes are expressed in the unfertilized egg, and they make maternal mRNAs. And remember, that's what's in the oocyte already. The maternal mRNAs are going to stay dormant until fertilization. No need to be making any gene products or any proteins because, well, there's nothing to do. The maternal mRNAs provide most of the proteins that are needed in very early development until that blastoderm is formed. Then we have the segmentation genes, and these direct the formation of the right number of body segments. So if you have too many or too few, then there's going to be some things wrong. And these are transcribed after fertilization. Again, you're not going to segment anything if you're not fertilized. There are different subclasses of segmentation genes. We're not going to go over these, but if you're interested, you can certainly look at it in your book. Then there's the homeotic genes, and these specify which organs and appendages are going to develop in a particular segment. So after segmentation genes are expressed and you've got your segments, then the homeotic genes kick in and say, okay, I'm going to be making the wings right here in segment 4. All of those things have to be coordinated spatially and time-wise because you can't make an appendage in a segment that doesn't exist, right? So you can do experiments with Drosophila eggs and look at the distribution of these different maternal gene products. And if you look at bicoid, which is a protein that's involved in developing the anterior and posterior sides, so kind of the polarity of the cell, you can actually look at the concentration of this protein in a normal egg. And that's what this dark part is, where all of that protein is. And as you get closer and closer to the posterior end, you don't see anything. If you remove that, then you're going to get a very different shape. And that mutant is actually not going to develop very well. So if you look at the double posterior larva here, you don't have an anterior, so you don't have a head. And that's bad. So you just kind of, this is not going to survive. You're not going to create a living fruit fly. So all of these different segments have different proteins at different times, different places, and it's really important to coordinate all of these things to create a functional organism. So this just goes over kind of what we were talking about, that this protein is a major anterior morphogen. So if you don't have it, you're not going to develop the anterior portion properly if you're a fruit fly. It acts as a transcription factor and a translational repressor. It's actually, the mRNA is synthesized by the nurse cells, which help to mature the oocyte. And it's deposited near the anterior pole in the egg. So this is already predetermined in the oocyte before fertilization. Then the protein diffuses through the cell to create a concentration gradient that radiates out from the anterior pole. And it goes from being really concentrated at the anterior to non-existent at the posterior. Hox genes are responsible for development of structures in defined body parts. So Drosophila has one Hox gene cluster. Humans have 4. But the Hox genes help to develop all the different structures, like the wing and the abdomen and the thorax, for a fly. Translational repression regulates developmental pathways in a major way. Many mRNAs are just hanging out in the egg way before they're actually needed. So they have to be repressed until it's their turn. And if they're not repressed, then you can have major problems. And that leads us to talk about stem cells. Lots of debate, ethical debate about stem cell research. Not what we're doing here. We're just talking about stem cells and what they are and why they are so desirable to understand. So a stem cell is a cell that can differentiate into various different tissues. There's totipotent cells that can differentiate into any tissue or a complete organism. So these are like the creme de la creme of stem cells. Then you've got pluripotent cells, which can give rise to all of the germ layers and many different tissue types. But they can't differentiate into a complete organism. They can only help make all the pieces. Then there's unipotent cells, which can develop into one type of cell or tissue. So when we look at the development potential of stem cells, when you have fertilization of an oocyte, you create the morula, which is a complex of totipotent cells. So they will develop and give rise to all of the things to make a whole person. When you make a blastocyst that has all of that, the totipotent cells, you're going to have some pluripotent cells that are created that can differentiate into the different tissues. And that's going to generate your circulatory system, your nervous system, your immune system. But those pluripotent cells can only differentiate into the tissues. They can't create a whole human being. So you can't just say, well, let's just start with some pluripotent cells and make a person. You can't do that. They don't have the blueprint. Stem cell proliferation versus differentiation and development. There are kind of two different processes that are going on at the same time. So stem cells have to proliferate to renew themselves, right? But they also have to differentiate to replenish the different cell types. So they have to balance both. And depending on the time and the place and the body's need, the organism's need, the tissue's need, you can either get kind of an equal mix of self-renewal and differentiation. You can get kind of sectional differentiation, or some kind of a gradient to go from, okay, these cells are self-renewing, and then as you get to this part of the tissue, then you've got more of a mix of self-renewal and differentiation, and then these stem cells are only for differentiation. So when you hear about research with stem cells, if you just hear stem cell, that's way too broad of a term. And hopefully this conversation will help you to, take what you hear and really process it for what it is versus what some of the hype may be in the news media. You know how that can be. So embryonic stem cells are pluripotent cells of the blastocyst, and these are the ones that are used in embryonic cell research. These are the ones that have a lot of ethical gray area surrounding them because you have to destroy an embryo to get them. This is not an ethics class, and I am not here to tell you that people should or should not be doing any type of research. But I'm trying to help you be a more informed human being so that you can make those calls for yourself. Then there are adult stem cells. And these are more limited, okay? They can't do all the things that embryonic stem cells can do. They're considered multipotent, but they are not pluripotent. What that means is that they have a niche, okay? There's a microenvironment that adult stem cells will say, hey, I'm going to hang out here. I'm going to divide. I'm going to self-replenish. And maybe sometimes they'll differentiate too. But it needs a very special microenvironment to do that. And it also requires, as part of that environment, signaling from neighboring cells to tell it, hey, you should keep dividing. And understanding all of this signaling and understanding how to harness this in the lab to have what's called immortal cell lines has been a great help to the research community, because we can use these cell lines to do research. And it's expensive, but it's not as expensive as expending human life. So while it may sound a little bit weird to have these immortal cells that will just keep dividing, and there are, of course, caveats with using these things, because that's not what a normal cell does. But it at least gives us some insight as to how the different systems in a cell work, so that we can apply them to larger and larger organisms and be, a bit more educated in the things that we try when it comes to formulating drugs and all kinds of treatments. That's it for chapter 28, and that's it for the course. Last lecture, woohoo! You can celebrate, that's alright, I won't be mad at you. Make sure that if you're a little bit iffy on some of the protein structure conversation, that you revisit chapter 4, and that you try to take the information from this chapter and kind of intermingle it with DNA, RNA, and protein metabolism, because all of these chapters go together. And then, once you do that, think about the techniques that we talked about in chapter 9 on how to get some of those things done. How do we study the systems that we're talking about in chapter 28 using those techniques? As always, stay safe, and I'll see you in class.